The title of this talk is Challenging and Promises for Universal Influenza Virus Vaccine. I guess there's no need for me to elaborate how important, if it's all possible, to develop a universal vaccine, either for seasonal flu or for pandemic flu. What I'm going to do today, actually, we're going to share some of our own work in an effort to develop a universal vaccine. This slide is just a cartoon to show that flu virion, there are some conserved antigens on the surface, like M2, neuraminidase, HA2, actually there's HA0, yeah, HA2 or hemagglutinin, which is more conserved. And there are also some internal proteins like nuclear capsids, okay? The highlighted here, M2, MP, and HA2 are the examples I'm going to share with you today. The first example is our investigational universal flu vaccine, which is based on M2 peptide and also hemagglutinin cleavage site peptide. It's a bivalent vaccine conjugated to meningitis outer membrane protein complex called OMPC. The vaccine M2 and from flu A and HA0 from flu B. Vaccine is formulated in Merck alum or alum plus saponin-based adjuvant iscomatrix from CSL. This is just show you the real sequence of this vaccine component. M2 is extracellular domain, only 24 amino acid, which is very well conserved. Okay, so if you raise antibody against, let's say, Hong Kong 68, this peptide will cross-react with many of the strains. Does not cross-react with avian flu M2. Okay, thank you. Now, there's no counterpart for flu B for M2. There's, so in order to develop a vaccine which can cover both flu A and flu B, you need to have a B component. For that reason, we have identified a region which is the cleavage site of hemagglutinin. Shown here, as you see, this is a cleavage site, it's exposed loop, which is almost 100% conserved in flu B strains. Okay. So here I show you the immunogenicity and protection of this bivalent vaccine in the mouse challenge model. Do the dose escalation here. On the right side is antibody titers. On the left side is the protection against the lethal challenge. You can see the vaccines are rather potent. We're talking about a nanogram of antigens. Actually, if you look at against flu A challenge, 50% protection, and the flu B challenge, 50% protection. The dose required is a range between 3 nanogram to 30 nanogram. I think they did not use my slide I gave to him this morning. You extracted from my PDF file, which was given to you quite a while back. Do you have a slide set that I have this morning? Not a PDF, it's a PowerPoint slide.
The second from the top on the left. Yeah. Right. The second from the top on the left. There you go. Yeah. No, the second.
So this is our clinical data from phase 1 trial. In this case, the vaccine is 5 microgram peptide plus adjuvant alum alone, or alum plus low dose iscomatrix, or medium dose iscomatrix, high dose iscomatrix, which represent 20, 60, or 100 microgram iscomatrix. Okay, this shows the antibody response against the M2 peptide with The alum alone, formulated with alum, actually induce very low titers. When you add ISCO matrix, the titer is significantly enhanced. It appears that different adjuvant levels give you different levels of responses. We also look at antibody response against HA0. This is slightly different from response against M2. You see after single immunization, the antibody shows up very fast. And there's no dose effect with regard to ISCO matrix. This could be that these individuals might be primed already for the HA0 antigen. So in summary, that the bivalent peptide conjugated vaccine is immunogenic in humans. Safety profile is actually related with the total antigen dose, OMPC, primarily the carrier, as well as the adjuvant. At the PD2 level, after post-dose 2 and forward, the titers with ISCO matrix adjuvant is like 10-fold higher than without ISCO matrix. The antibody level corresponds to the levels that you would see in the mouse challenge model. This antibody, if it were the same titer in the mouse model, it would be protective. Okay. Now let me switch very briefly to the second example. That is using recombinant adenovirus expressing nucleic capsid protein. As one of the panelists mentioned before, adenovirus is very good to present antigen, elicit T-cell responses. So in this case, you can see on the left panel is CTL response or CD8 response. The mouse were immunized with a different number of particles, 10 to the 4th to 10 to the 8th particles, and measured against splenocytes or measured for responses for a CTL epitope or a peptide pool. You can see there's a very nice dose response. Actually, the response is quite robust. Now, if you do the same, in the parallel, you have another panel of mice immunized similarly, then you will challenge these mice with a lethal dose, okay? In this case, we include M2 as a control. As I showed you before, M2 gave you 100% protection. The animals immunized with adenovirus expressing MP actually even gave you very robust T-cell response, do not give you 100% protection. Okay. So relatively speaking, on the experimental system, M2 is more potent than MP. So we did a second experiment, that is whether if we combine M2-based vaccine and adenovirus-based vaccine, would that enhance potency? So in this case, we challenge animal with 100 LD90. So the M2 alone here, when you challenge it with this high dose, the M2 no longer protect 100%. However, if you add M2 together with adenovirus, you bring up the protection efficacy, eventually achieve 100% protection. So there's some kind of additive effect between M2 and T-cell-based vaccine. Now I'm talking about HA. There's more recent data, HA2-based approach. As you probably all know that for the last couple of years, the HA2, there's monoclonal antibodies, human monoclonal antibodies identified which neutralize broad stream of flu viruses. Most of these antibodies are actually confined to the group 1 virus, including H1. More recently, actually just the last couple months, Peter Palese in Monsanto Medical School identified a mouse monoclonal antibody, which recognize group 2 or H3 virus HA2. So the way of design HA2-based antigen, because HA1 is the dominant antigen. So what you want to do is remove the H1 so that immune response will be directed against the H2. However, HA actually is metastable during viral entry. When this virus gets to the endosome, exposed to low pH, HA will have a dramatic conformation change. So in the past, people simply express HA2 antigen, they automatically assume a post-fusion conformation, so which is not what we want. So the idea is that if we can develop a approach or approach somehow to fix HA2 antigen in its pre-fusion status. The way we do this is called the minimization protein design. Basically, based on the crystal structure, HA3, using a computer algorithm, we can identify the minimum contact residues of HA1. Those are the residues you want to keep together with HA2. Then remove most of the HA1 sequences. When you do so, you expose some hydrophobic residues, which you would like further mutate. Those residues make it not hydrophobic. In addition, to stabilize HA2 structure, we introduced additional mutation in the HA2 sequence. So as a result, we generate a molecule like this, HA2 with a small number of HA1 residues, which is linked all together by linkers. So this molecule is expressed in E. coli. When you do the sizing column, the molecule and it loaded up like 84 kilodalton, which is consistent with the predicted trimeric form of HA2 molecule, 90 kilodalton. This shows the CD spectrum. At the neutral pH, the CD spectrum is consistent with the predicted value. Now, normally when the full molecule HA, if you switch to low pH, you will increase helicity. In this case, we do not see the increased helicity. On the other hand, actually reduce helicity, which again is consistent with the notion that this molecule is in the status of neutral pH. To further probing for structure, we made two additional mutants. Basically here, introduce a pair of cysteine adjacent to each other when the molecules in pre-fusion status, okay? Once, if these will form disulfide bond. However, if it's in the low pH status, these residues will be like 90 angstroms apart, so they won't form the disulfide bond. So we look at the oxidation status looking for ion. It did turn out actually these all seem to be in oxidative status. That again support the notion that we do have a molecule which is in the neutral pH form. So with the help of Peter Palese, we were able to get the monoclonal antibody 12D1, which as I mentioned before, we're looking to see whether this monoclonal can bind to the HA molecule that we just produced. So here, this is the antibody concentration, simply OD numbers. Okay, use a control peptide, there was no binding. Just highlighted here that monoclonal antibody is capable of binding to the HA, recombinant HA2. Just like it also binds to recombinant hemagglutinin. This you see the protection study in the mice. Recombinant HA2, also those HA2 with the two cysteine pairs, okay? We use CpG as an adjuvant. So in this case, in the CpG adjuvant, of course, as is predicted, did not protect the mice, whereas mice receiving all three different forms of recombinant HA2 were protected against the lethal challenge. Now this is a last slide, so I want to show you some caveat with this so-called universal vaccine. On the left panel is the way mice challenged with different vaccines, M2, HA2, or the convalescent mice. Okay, oftentimes people show you the protection data against lethal challenge or mortality, not showing the weight change. So if this is a convalescent mice, when these mice receiving challenge, they don't really lose any weight. Actually, they gain weight. We do not have data here in this particular slide, but if you immunize homologous matched HA vaccine, this is what you will see, okay? Whereas the mice immunized with M2 or HA0, they lose significant weight. So they don't really achieve kind of same level of protection as matched vaccine. On the right side is that we repeatedly protective studies in the FC knockout of mice. So the top panel is passive transfer study. The serum from mice immunized with M2 or HA0 antigen. In the white type virus, you see this type of protection, complete protection. But in the FC knockout mice, the protection all reduced very significantly. The bottom panel is HA antigen. Again, in the white type virus, you have full protection, but in FC knockout mice, they all reduced in terms of protection. So in summary, that conserved antigens such as M2, HA0, MP, or HA are capable of conferring protection against lethal challenge in mice model. The protection may be enhanced by combining 2 or more antigens. However, in general, this protection is rather weak as compared to homologous matched vaccine. And there's no indication in our hands. We have never been able to detect the neutralizing antibody responses in these protected animals. M2 and HA0 conjugated vaccine given with a proper adjuvant are immunogenic in humans. Lastly, identification of conserved neutralizing sensitive HA2 epitopes established basis to pursuing HA2-based approach. Designing such a vaccine to elicit neutralizing antibodies, that would be the ultimate goal. If we can generate a neutralizing antibody, it's wonderful. However, that might not be easy, may not be a challenging approach. Okay, this is all the contributors. Many people contributed to the program. Would like to thank Rex from Indian Institute of Science who designed this HA2 vaccine, also Peter Palese for providing us with monoclonal antibodies. Thank you.